All right. I was asked a question by your colleague over here that the MHC molecules are primarily involved in presenting peptide antigens. MHC class 1 can hold smaller peptides, 8 to 12, sometimes 14 amino acids long, and MHC class 2 can hold larger peptide sequences, somewhere between 14 up to 18 or 22 amino acid sequences. Now, when your antigen presenting cells are only able to present antigen, which is a protein breakdown product, a sequence of amino acids called peptides, to activate T cells. That means the T cell receptor can only see peptide bound to an MHC. Whether it is presented through MHC class 2 to activate CD4 T cells or through MHC class 1 to activate CD8 T cells. Now the question was very integral and important, and we were going to discuss it later, but I think it's pertinent here to discuss it as well. And the question was that how do B cells that respond to three-dimensional antigens, and the three-dimensional antigens can be lipids, carbohydrates, nucleic acids, and proteins. How do they actually interact with them, right? So what you have here is a typical pathogen, right? And the pathogen in its whole construct would have protein-based antigens in it, lipid-based or carbohydrate lipid-based antigens that could be a part of the membrane, or could be cell surface proteins. Regardless of the kind, you can have all three antigenic components present in an extracellular pathogen that comes in. When it comes in, two steps take place simultaneously. The antigen presenting cells phagocytose the pathogen, break it down, and present the antigen, but only the protein antigen, that is primarily going to be the peptide sequence, MHC class 2, and the correct naive T cell will come and recognize this peptide bound to the MHC and go through a series of clonal expansion to become an effector helper CD4 T cell. While this is happening at one end, at the same time, the B cells are engaging the cognate antigen that they would on the pathogen using the B cell receptor. Now the B cell receptor can directly engage a 3D structure. It does not need antigen presentation per se. And when it does, this B cell can directly get activated, of course, goes through clonal expansion and becomes a plasma cells that start secreting the initial IgM, and we keep saying that's the hallmark of an initial inflammatory reaction, that your blood levels have a surge of IgM, pentameric form, that kicks in. As yet, this B cell has not received any T cell help. While a cluster of B cells expand clonally and do this, another group of B cells which have the same B cell would actually engage the pathogen, and using the B cell receptor, which is bound to the antigen and therefore the pathogen with it, not shown here, okay, it would endocytose or downregulate its B cell surface receptor, dragging with it the pathogen inside. Once it does that, again, just like the regular phagolysosomal functionality, this pathogen inside the B cell is again broken down. And what are you left with? You're left with small cleavage products of lipids, proteins, and carbohydrates, all as a mixture, including the B cell receptor that's been degraded in the phagolysosome. At this stage, the MHC class 2 molecule comes in, and this MHC class 2 is now going to be selective because only peptide sequence gets loaded onto it. And it gets the peptide loaded onto it and goes onto the surface. Now, what you need to bear in mind, and this is the specificity of the immune response, is that the same peptide sequence that this T cell got activated against went through clonal expansion, and now is an effector CD4 T cells. Effector means it is able to respond. Naive means it is not able to respond, it's able to get the signal internally. So here the T cell receptor is used to activate, go through clonal expansion and become an effector T cell. The same T cell receptor here, because it's a clone, it's an exact copy of that. The same T cell receptor will now recognize the same antigens. They have to be the same antigens. Okay? And once this CD4 T cell recognizes the same antigen, now you will have CD40 ligand rearranging with CD40 on a B cell and release of cytokines that will act on the cytokine receptors, and then they will go and act on the DNA to introduce the class switching. And now this B cell can go again through the process of clonal expansion while it is still engaging the antigen on the surface. So this still needs to be there. Signal 1 is still there. And this now becomes signal 2, which is CD40-40 ligand interaction, and signal 3 from the cytokines to go back to the genome of the B cell and induce class switching. Depending on the kind of cytokines, the activation-induced cytidine deaminase that is activated through CD40-40 ligand will be told to act on switch region 1, and the next switch region could be any depending on the kind of cytokine, right? If the cytokine, for example, was transforming growth factor beta, it will act before alpha 1, and you will have a B cell now that can again go through two processes to become a memory B cell and induce becoming a plasma cell secreting IgA. Okay? Is this clear now?
Just a little point. What determines which cytokine is secreted?
The type of cytokine secreted here is determined by the macrophage inducing the helper type, and the macrophage that induces the helper type is educated by the kind of pathogen it engages, because the pathogen engages the particular toll-like receptors on the macrophage, telling it, hey, I have a lot of lipopolysaccharide, or I have a lot of single-stranded RNA, therefore induce a different type of T cell and go back to it. Okay? So that's the simplistic baseline. Yeah. So the toll-like receptors on the macrophages that are there, to be more precise, let's not just say toll-like receptors, let's say pattern recognition receptors. Because in addition to toll-like receptors, you have another cluster of NOD and other receptors which are similar. They recognize molecular patterns of conserved molecules which are found in pathogens but not in us humans. These molecular patterns need not be antigenic in their nature. Okay? Or they can be antigenic, does not matter. Okay? So, for example, if a bacteria has that flagella, it will induce the engagement of a different toll-like receptor as compared to if it's a virus that has got a single-stranded RNA. Okay? Therefore, depending on the kind of toll-like receptors that are activated, the macrophages or the dendritic cell, regardless which antigen-presenting cell it is engineered to give rise to different set of cytokine signaling to activate this T cell. Okay? So while it's activating it, it's also giving cytokines, telling it to differentiate into a certain type of T helper or another. Okay? It depends on what is required. You get it? Now, having said that, it is not always true that that should happen. You know why? Because cytokines are, unlike PCR and MHC, which is cell surface protein engaging a cell surface protein, cytokines are environmental signals that are there. So these cells and these cells both have a lot of different cytokine receptors. So the most dominant cytokine that is there, which should be by logic the one that is being produced by the antigen presenting cell, will signal the naive T cell to differentiate into one or the other cell type. You get what I'm saying? But artificially, you can have a cytokine here that is being produced in that environment that is more than what is being produced by the antigen presenting cell, and then the outcomes can be different. So you want a T helper that should, for example, give rise to a T helper 1 response and therefore target viruses or intracellular pathogens. But if the environmental cue is strong enough, it will end up making a T helper 2 cytokine or T helper 2. The cytokine will dictate it to make a T helper 2 cell, and this will induce allergic responses. It will induce mast cell and IgE production. You get what I'm saying? So although it is designed to do something, but cytokines can be manipulated by the environment. Okay? So does that make sense? Relatively simple? Okay. Now, we just go back and reflect on where we stopped yesterday. So we see that if anything is extracellular in origin, it is taken up into the endosome and the phagosome, phagolysosome, gets loaded on MHC class 2 and goes on the surface. And if something is cytoplasmic in a cell, it will be broken down, the protein will be broken down by the proteasome. The protein can be malfolded protein or a protein that is not wanted by the cell anymore. It could be a cell surface receptor that needs to be downregulated. Regardless of what is the causality, once it is gone to the proteasome, chopped down into different peptide sequences, they are actively transported. They use energy-dependent mechanisms to pump these peptide sequences within the endoplasmic reticulum where they get loaded onto MHC class 1, and then the MHC class 1 comes onto the surface. Now, if you recall, we discussed earlier that the role of the T helper cell is, of course, what we just discussed over here, to provide help to B cells, right? But another role also for these T helper cells is that you have naive CD8 T cells. Okay, let's pick up an example of the external pathogen to be a virus, right? So let's say that this thing was internalized as a virus, and now the signal goes more to the B cell to class switch, and now it is going to secrete IgG, okay, antiviral antibodies, and they will be targeting the viral coat protein. In the same time, you want the CD4 T cells to come and help CD8 T cells, which are naive, to go through clonal expansion after they get antigen presentation, and this help of clonal expansion is provided by the helper cell here for this cell to actually become now a cytotoxic T lymphocyte. And what do cytotoxic T lymphocytes do? They go back and kill the virus infected cell, right? So in case of HIV, which is the cell that is infected? Say again? CD4. So this cytotoxic T lymphocyte is going to go back and kill which population? The CD4 T cells. Okay? And when it does that, it starts over time getting rid of all the help. And help that is specific to the pathogen. Right? That's why at the end of the HIV, the person, when it actually progresses to AIDS, when the cell count has gone low enough, the person is not dying because of HIV. It's dying because of environmental pathogens that you cannot defend against anymore because of the lack of help both for B cells and CD8 T cells. Okay? Does that make sense? Now the key question is that if the pathogen over here is a virus, and we say that it needs to be cytoplasmic, right? For it to be expressed on MHC1. So this antigen presenting cell is expressing something on MHC class 2, and the antigen presenting cell is also expressing something on wrong MHC, sorry. It needs to be MHC class 1. Okay? So for an antigen presenting cell to present antigen both on MHC1 and on MHC2, this whole structure, this whole pathway is a dilemma. Why? Because if there's a bacteria, extracellular, phagocytosed, it'll go to MHC2. Right? So if you talk about a poliovirus, right? This antigen presenting cell will see a viron going through the bloodstream, phagocytose it, present it on MHC2, and it's good enough to activate the T helper. How will it activate the cytotoxic T cell that is actually supposed to go and kill the virus infected cell? You get the point? It cannot do that because the poliovirus is not directly infecting the antigen presenting cell, and therefore the viral proteins are not a part of the cytoplasm of the antigen presenting cell. Therefore, MHC presentation will not happen if only these pathways existed. You get what I'm saying? And vice versa. So what you have in those contexts is something called as antigen cross presentation. Antigen cross presentation is a process by which antigens that should normally go through MHC2 pathway or antigens that should normally go to MHC1 pathway are crossed over. And how does it happen? It's particularly your dendritic cells that have this capacity to do more. Less is so found in the macrophages. And what typically happens is that the pathogen or a dying cell, whatever, which is phagocytosed, post-phagocytosis, the phagosome goes through another phase of maturation. So the phagosome, if you recall from yesterday, what is the pH in it? Low, acidic pH, right? After it's degraded things. And because of its low pH, there were two outcomes. One was things getting loaded to MHC2, okay? And other thing not shown here is whatever is left over is just thrown out of the cell, excreted out like feces. But in these particular dendritic cells, what happens is that once the phagosome is formed, degraded, broken down everything here, the pH that had fallen is now reversed. The pH starts to be pushed up back to the cytoplasmic pH level, and the phagolysosome then disintegrates. Once it disintegrates over here, all the contents that were inside the phagolysosome are now cytoplasmic. Once they are cytoplasmic, they will now go through the transportation of MHC class 1 pathway and therefore be presented onto MHC class 1. Go through the endoplasmic reticulum and there they go. Okay. Now, another less common pathway is that sometimes MHC1 can also directly engage over here, get an antigen loaded and go to the surface. But that is a very rare phenomenon, not noted commonly in infections. Okay. But the common pathway is that you have the proteins, degraded product thrown down into the cytoplasm and then into the endoplasmic reticulum and then they go. Now the similar process takes place for MHC class 2. Now if there is something in the cytoplasm here, let's go back, think about the macrophage and think about a macrophage that has been infected with HIV, for example. Now, because this macrophage is infected with HIV and it has not, let's say, phagocytosed another cell that is dying because of HIV, this still needs to present to MHC class 2. Stressed cells, what do? There's something called as autophagy. Ever heard of this before? No. A Nobel Prize went by, and thanks to the Arab population, we don't fast and we don't think what happens in fasting. So when you're fasting, and you're not eating for quite a number of hours, what starts happening is your own cells, for the purpose of energy requirements, they start eating themselves up. Okay? That's the first 10 days of Ramadan. After that, will become different. Okay? What starts happening there is the cell that is stressed due to lack of, either due to environmental stress or due to lack of energy. What these cells do is they send a bud off of their own cytoplasm outside. Okay? So they will send a bud out of their cytoplasm outside, make a small exosomal vesicle, release their cytoplasm and everything as a vesicular body, and then eat it back in and degrade its products down. Therefore, this eating its own cytoplasm or eating its own contents by the cell is called autophagy. Auto is self, phagy is eating. So the cell starts to eat its own cell, and when it does it, anything in the cytoplasm, any protein that was in the cytoplasm, if you recall, here, these cytoplasmic proteins. So these cytoplasmic proteins will be thrown out and taken back in, and then goes through the process of phagocytosis. And anything that is phagocytosed goes on to MHC class 2. So there it comes here and gets onto the surface. So any cytoplasmic product in an antigen presenting cell is therefore, through the process of autophagosomy, put on the cell surface. There are different types of autophagy. There is something called as microautophagy and macro. What I just described is macroautophagy. Macroautophagy will have double membrane. Why? Because when the cell makes its own projection outside and sends off a bud, it will have one vesicle which is bound by the same cell membrane. And then when it takes it back in, it will have another membrane on it because of the phagosomal membrane. So this is a double membrane macroautophagy product that is formed. The other is microautophagy. What cells do is, instead of budding out a whole part of them with the exoskeleton and everything, what they do is they start aggregation of small lipid layers inside. So lipids start to accumulate in an area of cytoplasm and then form a vesicle. Okay? And when they form a vesicle, whatever is inside is the cytoplasm. And that is microautophagy. Micro will only have single lipid vesicle forming and containing the cytoplasm plus the contents of the cytoplasm. That is again treated as a phagosome. Again gets loaded to MHC class 2 and therefore onto the cell surface. Okay? Was this clear? Easy, simple, not too difficult? Yes.
What is the purpose?
Good. Normally cells do this under stress to recycle their products. But in the context of antigen presenting cells, it is a way to express a cytoplasmic protein that would by normal process only go to MHC class 1 to actually go to MHC class 2 so that it can activate the CD4 T cells to become helper subsets to come back and give help to CD8.
So in the case of, for example, viruses, the sum of the cytoplasmic content will have the virus antigen.
So if a virus is infecting my respiratory epithelia, then the dying epithelia, respiratory epithelia, will be phagocytosed by a macrophage and put on the surface of the MHC class 2 molecule, only activate what? CD4 to become a T helper. What about cytotoxic T cell? How will it go to MHC class 1 if it's phagocytosing? Exactly. So it needs to do cross presentation of a phagocytosed viral particle to actually induce activation of CD8. On the other hand, if you have the pathogen which is supposed to be targeted by the macrophage in the opposite direction, right? So if you had if a virus itself, so there are many pathogens, let's say Mycobacterium tuberculosis, okay? TB or Listeria itself go and infect the macrophage, right? And they are Listeria in particular is cytoplasmic. So when it is cytoplasmic, by default, you have MHC1 expression for cytotoxic CD8 T cell, but you have no expression for MHC class 2. That's where it needs to carry out either micro or macro autophagy to push it to the MHC class 2 pathway and therefore on the surface. Okay?
Intracellular antigens.
Yeah, cytoplasmic intracellular antigens. Intracellular is everything.
Does cross-presentation take place every time an dendritic cell phagocytoses a pathogen?
Now, the exact key signals are not known. Sometimes it doesn't happen. It happens and sometimes it doesn't. They have not been able to make an association with pattern recognition receptors yet. But when it does, when it doesn't, is not really known. But what is known is there is a subset of dendritic cells which are called as plasmacytoid dendritic cells. They usually are more in circulation. They are more efficient at carrying this process out than the others. Okay? Right. Yeah.
In microautophagy, how's the internalized cytosol treated as an extracellular antigen?
It's not treated as an extracellular antigen.
Because it's
So you see,
expressed on MHC2.
I'll try to explain here. Macrophage, when they get stress signals, they start carrying out process of autophagy. One of them is what you're talking about, microautophagy. They start making lipid bilayer, they start assembling lipid bilayer vesicles in their cytoplasm. So what you essentially have is a small vesicle formed with the general cytoplasmic contents that are there. Now, because the stress signals push this macrophage to use this vesicle for energy purposes, okay, it's eating its own self. You have other proteins that assemble on the surface, pH starts to drop, lysosomes come and fuse, and they end up breaking down all the cytoplasmic contents. And the objective is that it needs to use all of this for its own self or the environment or neighboring cells, right? So while it is doing it, you have from the endoplasmic reticulum MHC class 2 come here, load and go to the surface. Okay? So this MHC is sort of a byproduct of these pathways.
Doctor?
Yeah?
So for cross-presentation to MHC class 2, you want signals?
That is what is not clearly known. Because the processes of macro and microautophagy are very evident and clearly visible in cell stress. Even in cells that are not phagocytic cells, epithelial cells, endothelial cells and others, when they are stressed, they start carrying out autophagy. Hepatocytes carry out the same. Okay? So it's a common pathological feature that is observed in cells when they are stressed. And they also observe the same process taking place in macrophages. Now, whether there is a real stress signal or there is not a stress signal, they were not able to identify that yet. So there's a lot of work going on it as we speak. Okay? So, but this kind of a vesicle, okay, is a single membrane phagosome because it is a single membrane assembly inside. If it was a bud off and taken back in, that would be the double membrane, and that's typically macroautophagy. Right. Now you have the big picture in your mind. We're going to focus on each small subset and try to discuss them in detail. Now, what happens when a dendritic cell actually sees a pathogen, right? So you have a pathogen here, which is of course expressing the pathogen associated molecular patterns, and they will be recognized by pattern recognition receptors like dectin-05 or the toll-like receptors. Now notice this immature dendritic cell is primarily found in the peripheral tissue. So there's a difference between the antigen presenting cells in them being immature and mature. Notice we are not using the term naive and effector. We are not using the term naive and antigen experience. We're just saying immature and mature. The difference is that an immature cell likes to sit in the tissues because that's where the greater probability is that it will see the antigen or the pathogen. And once it matures, it is primarily moving back to the lymph nodes. So that means some cell surface signals or molecules would change. Some would tell it to go and sit in the tissue, and the other would tell to go back and move towards the lymph node, right? And these kind of signaling are based on chemokines, okay? Chemo is chemical, kines is kinesis, movement. So chemical gradient based movement makes cells go from the blood to the tissue, back to the lymph node, and vice versa. They keep circulating around based on that. Now, the signals that are provided are already there. The tissue keeps providing a certain signal that this is called tissue, lymph nodes do the same, and circulation does the same. What differs are the chemokine receptors. You get what I'm saying? So think about it like this, right? You have a naughty little child in the back of the car making a lot of noise for a toy, right? And the parent says, you're going to be quiet and we're not buying the toy. What does the child do? Screaming, okay? Throwing a tantrum. Now, what is the child trying to do is block the signal that the parent is giving. Just does not want to listen to that input. Therefore, starts creating an alternate signal, right? Similarly, the cells, when they want to move from the tissue and go back to the lymph node, the parent is still saying you're not getting the toy, but it starts changing its behavior. So they start either down-regulating certain receptors or up-regulating certain receptors. Now, tissue-resident immature dendritic cells, they primarily have very low amount of co-stimulatory molecules. You remember we talked about CD80 and 86 as a co-stimulatory molecule yesterday? Immature cells have got very low amount of that, number one. Number two, their capacity to phagocytose and antigen present is low. So they do not do it at a higher pace. Number three, they like to remain in the tissue. Now, they have phagocytic receptors like DECTIN1, DEC205, Langren, and DC sign. And notice DC sign is one of the receptors that primarily HIV uses to access tissue-resident dendritic cells. Okay? Now, the chemokine gradient that is keeping them there is CCR1, 2, 5, 6, and CXCR1 and 2. They keep them here in this particular environment. Once they get activated, once the toll-like receptors are engaged, they get activated. There are two very significant features. A dendritic cell sitting in the tissue is lowly phagocytic, meaning it is just phagocytosing at a very low rate, and the reason why it's doing it is it needs to sample its environment. Like right now, you're sitting here and you're sampling your environment. Your body is telling you what's the temperature around you, what is the noise around you, how much light is there. Okay? You may not be cognizant of it, but it's there. You're sampling your environment. Dendritic cells do the same. Low-grade phagocytosis. But the moment their toll-like receptor or DC sign or DEC205, any of these receptors that recognize pathogens, the moment it is engaged, they stop further phagocytosis. They do not phagocytose anymore. And why? Because they need to express on the surface the pathogenic antigen as compared to the self-antigen. Do you understand that? So when they're carrying out regular phagocytosis, when the tissue is normal or normal apoptotic cells are there, they're just taking small chunks out of them. They're presenting on the surface over here an MHC class 2 self-antigen. Right? When they will phagocytose, they will eat something. When they will eat something, it is your own cells, your own tissue. And when it is phagocytosed broken down, it's going to come on as your self-antigen. The moment their pathogen-associated molecular patterns engage the pattern recognition receptors, or the toll-like receptors, to put it simply, they stop further phagocytosis. Because now they've engaged the pathogen. They do not want to dilute the antigen of the pathogen to be expressed on the surface. That is the first change that happens. The second change is they increase or upregulate the surface expression of MHC class 1 and class 2, meaning more of the pathogen will be now loaded onto the surface. Third, they start migrating back towards the localized lymph node. And while they are migrating to the lymph node, that's when these changes are happening. They're increasing more expression of MHC class 1 and class 2. They're also increasing the expression of CD80 and 86, which is co-stimulatory. And they end up going into the targeted lymph node. While they go into the lymph node, that's where you have your T cells. And T cells require 3 signals for actually getting activated. The first one is the T cell receptor of the T cell. It needs to engage the peptide that is bound to the MHC that the antigen presenting cell is now bringing. Okay? The second signal is the co-stimulatory signal that I was talking about. You need to have CD80 and 86 on the antigen presenting cell, on the dendritic cell. That will engage CD28 on the T cell to give it full activation. And the third signal we need are cytokines. The cytokines that will tell this T helper to become, this naive CD4 to become what kind of T helper, and the cytokines required to expand CD8 T cells. Right? Yeah.
You said yesterday about the T cell recognized MHC and the antigen presenting cell.
Yes, so the T cell will recognize the peptide bound MHC on the antigen presenting cell. Which receptor? So the T cell, okay, good. So the T cell receptor will first recognize the MHC and then the peptide that is held by the MHC. We discussed that yesterday. Okay? But I'm trying to simplify things. If I keep repeating those details, other details will be lost. But you're right. That's the sequence. So the first sequence is excellent. Let's talk about that now. Now, because that is happening, this graph is showing you the inflow of T cells to the lymph node, okay, and the outflow. Now, the T cells that you have are in circulation, 2 parts. They are moving in your blood. From the blood, they move back to other lymph nodes or secondary lymphoid organs, and from the lymphatics go back to the thoracic duct, back into the circulation. So for all practical purposes, your B lymphocytes and T lymphocytes are found in 2 places, in circulation and in the lymphatics. Now, if you think about it, when the antigen presenting cell is there, for example, let's say that I am a doctor working in the hospital and I get a needle prick injury after taking a blood sample from an HIV positive patient. Okay, yeah, bad. That happens. People were stupid trying to recap. Very common things that happen in hospitals, right? So the injury takes place on my finger here, okay, and the HIV virus would enter the localized tissue. From here, the antigen presenting cell is going to the localized lymph node, which is supratrochlear, and then to the axillary. And that's where it's going to sit, trying to wait and activate the CD4 and the CD8 T cell. While it is moving from the tip all the way here, it also matured. It was immature and tissue resident. Now it has matured, it is presenting the antigen and the co-stimulatory molecules. While it is doing that, the T cells that are specific to actually target HIV may not be in my axillary lymph node and typically on the left side. They may be circulating somewhere else. So what happens is that you have a high level of input of the T cells going to these particular lymph nodes, and they keep moving on and on till the correct T cell enters. The moment the correct T cell enters, then they don't exit anymore. Their levels drop. This is time, this is the number of antigen responding T cells. They stop and they remain within the lymph node. This is where antigen presentation is taking place and clonal expansion is taking place. After antigen presentation and clonal expansion, signal 1, 2 and 3 when it's given, now a higher number of the antigen experience clonal expanded cells which are effector now leave the lymph node, and that's what's happening here. So you have naive cells coming in, getting activated in the lymph node, therefore they're not leaving, and once they are activated, now they go out. Now what happens at the interface of these two cell types? If you recall yesterday, we said these cells are like billiard balls. They're not adherent cells, they're free floating cells. So for them to actually engage, you need to have transient interactions between the naive T cell and the mature antigen presenting cell. T cells on their surface have got a low level of expression of lymphocyte functional antigen 1, LFA1, along with the CD2 molecule. While the antigen presenting cell have on their surface ICAM1 and ICAM2, which is intracellular adhesion molecule 1 and 2, along with CD58. So think about it like this, like if you're most likely in the shower, old people when they slip, it's just because the soap comes in between and they cannot hold on. So think of this as weak interaction. It's just like your shoe giving you grip to walk on the ground. It's not a fixed interaction, but it is an interaction that still has some amount of adhesion, so things don't just fly off. Okay? So your antigen presenting cell is sitting in the lymph node waiting for the correct T cells to come. If a T cell comes using LFA1 with ICAM1 and 2, it tries to engage. And once this engagement happens, it gives a little time for the T cell receptor and the MHC to come and align with each other, because they can be just spatially arranged in a totally different place. Okay? Now, based on that kind of engagement, this weak interaction allows them to align. CD4 will first recognize the MHC, then it will push the TCR to come in. Now the T cell receptor, like she very correctly identified, first recognizes the MHC1, then tries to recognize the peptide. If it doesn't recognize this, it moves away. But if the correct T cell comes with the correct T cell receptor that recognizes it, this interaction sends a signal inside back to LFA1, and this weak adhesion turns into a stronger adhesion. Now, please bear in mind, the T cell receptor interaction with MSC, this molecular interaction is not anchoring. It's not binding. It's just two things coming into surface with each other. Okay? So the thing that holds the T cell to the antigen presenting cell is the change in the LFA1 affinity because of the molecular signaling that takes place to bind to ICAM1 and ICAM2, keeping them in place. This is important so that it gives enough time for proper signaling to take place and the rest of the molecules to align. And what happens then is that the first signal of activation comes through the peptide MHC interaction through the T cell receptor, followed by co-stimulation that takes place through B77.1, which is also CD80. 7.2 is 86. So CD80 and 86 engage CD28 to give the second survival signal. Okay? And the third signal that is supposed to come released by the antigen presenting cells are a cluster of cytokines to tell this T cell what kind of differentiation it needs to form. Okay? So is this clear? So first is activation, survival, and then differentiation. That's how the three processes go. If all three are not there, the outcomes do not happen. Okay? And remember, LFA1 is the most important to keep things in place. Now, briefly, you remember a little while ago we discussed the majority of the signals here design what the outcome is going to be like. So what happens is, now here we are talking about only the third signal, the cytokine. So activation has happened, survival signal is given, the co-stimulation has happened. Now what do cytokines do? The cytokines, like if the antigen presenting cell makes a lot of cytokine like interleukin 12, this will push the naive T cell to differentiate after clonal expansion into a T helper 1. These T helper 1s make a product called interleukin 2, TNF alpha, and interferon gamma. Interleukin 2 is one of the core signals that is required for CD8 T cells to go through expansion. Okay? And interferon gamma, interferons are there to shut down viral functionality in the rest of the cells of your body. Okay? So basically interferons, if my body starts suddenly synthesizing a lot of interferon, across my body protein synthesis will shut down. That also means that if a particular group of cells are infected by a virus, they will suppress viral protein production, thereby suppressing viral production and viral replication. So that's what interferon do. TNF alpha. How many of you have fallen sick in your life? I believe everybody. So the feeling which makes you feel sick, right? That groggy feeling of being cold and not too well and crouching up like a mouse and not wanting to get up out of bed, not wanting to eat, feeling not wanting to, chat and talk, and your mood gets really bad. That is TNF alpha. Okay? So TNF alpha actually makes you feel sick, not wanting to eat. It also starts sequestering more iron into the liver, trying to decrease the amount of nutrition available for pathogens.
Is naming of it related to cancer?
Yes, it was originally identified there. Tumor necrosis factor. Okay, it was originally identified from there. Okay, but later they found out that its function is global. Okay, it also impacts fever production later on. So all of this, if you notice, interleukin 2, TNF alpha, interferon gamma, is there for viral infection. Therefore, whenever the antigen presenting cell will engage a virus, single strand, double stranded virus, it will induce, and the cytokine it will produce in a lot of amount will be interleukin 12, so that the T helper becomes T helper 1, and when it starts releasing its products, they actually help the T cells, they help generally to suppress viral functionality, and actually they also go back and affect the macrophages to carry out more effective phagocytosis. However, if the cytokine from the antigen presenting cell is interleukin 4 and signal 1 and 2 are the same, signal 1 and 2 are not changing here. Signal 1 is the peptide MHC interaction, and signal 2 is the co-stimulation that you get through CD80 and 86 engaging CD28. If those signals are still the same naive cell will now become a T helper 2. And this T helper 2 starts making interleukin 4, interleukin 5, and interleukin 10. These IL-4, 5, and 10 are actually going to help B cells to go through clonal expansion as well, but more so induce the production of IgE. And IgE is required for allergic responses, right? So if a pollen comes in, you will have the formation of the T helper 2 and giving rise to more humoral immune response per se. But notice one more thing. The products that are coming out over here, IL-10 is inhibitory for the T helper 1 path. And the products that come out here, interleukin 2 and TNF alpha, are now, and also IL-12, are inhibitory for the T helper 2 pathway. So initially, the cytokine may be a mix and a match. But whatever becomes the dominant cytokine in that environment dictates the immune outcome. Do you get it? And the products therefore come back and start influencing the pathway. So initially, the cells may not know whether they're becoming T helper 1 or T helper 2. But over time, when the cytokine signal becomes strong enough, they all start moving in one direction. Do you get that point? Yeah. So if you think about it in T helper 2, right? Some people in medicine, you will hear later, they talk about someone getting infected or having an allergic response, and then they end up having a viral superadded infection. Okay? So what does that mean? That the whole cytokine response shifted to Th2, right? And it was going through an allergic responses now. And now the person got infected by a virus, which could normally be managed by the T helper 1, but because the cytokines have shifted to the Th2, you get a superadded viral infection. Okay? Or vice versa. Some people start off with an initial flu-like symptom with a virus infection. You go to the doctor says you don't need antibiotics. And you go back home and say, okay, I agree, it's virus-like. 5 days later, you have high-grade fever, and you're going back and cursing the doctor again. Well, your immune system was playing games there.
Doctor, does that mean that in the presence of Th1, Th2 is going to be absent?
Yeah.
And vice versa?
Products. IL-10 blocks that. Induction. It blocks the induction. If they are there, they are there. But it will not make any more new come in. That's why it's the secondary effect. The superadded infections mean one infection process is in and another kicks in, but that is not being responded to because the immune response is not kicking in that direction.
Sir, but in the case of Th2, IL-4 will also suppress the protein synthesis, so it's still killing the virus.
IL-4, not much.
Not as the...
Not as far as interferons. Okay. Now, T cells are able to differentiate into a family of different effector cell types. You've got T helper 1s, which are induced by interleukin 12 and also supported by interferon gamma. You have the T follicular helper. These are the ones that give rise to different B cell responses. You have the T helper 2s, that of course we just discussed about, induced by IL-4. You have the regulatory T cells that are inhibited. They need to shut down the immune response. And you have T helper 17 that are known to cause chronic inflammatory responses. And interleukin 17 is one that is known to bring waves back of neutrophils in. So you know your immune response starts with the innate, shift to the adaptive, right? And neutrophils typically that are high initially in infection start dropping down while the adaptive kicks in, right? I know I'm talking rocket science now, but it's okay. And you're already brain dead. But what you need to understand from here is that when you have Th17 induction, neutrophils that should not come are re-recruited into it. Now, the balance is very small. Notice that if you have interleukin 6, if you have TGF-beta being made, the cell becomes inhibitory, regulatory T cell. But if you add interleukin 6 to it, it becomes pro-inflammatory. T helper 17 kicks in. If you swap TGF-beta, you still have IL-6 with IL-21, you start having T follicular help that induces IgG production later on. Okay? Sorry?
Both need to be present.
Yeah, both need to be present. Here, both need to be present. Here, only one. So, if you notice, this is a very simplistic diagram showing you what induces and what's the end product in terms of the helper cell. Here, I'm not showing the interactions they have with each other. The only interaction I showed was between T helper 1 and T helper 2 in the previous slide. What blocks and what doesn't. There's something you need to remember here are the transcription factors. What are transcription factors? After this I will stop. But you need to tell me what is in your mind, what you try to explain the term transcription factors as. Factors that are present in transcription. Let's modify it by saying factors that regulate and control sustained transcription. Meaning that as long as these are present within the nucleus, they will make a certain group of proteins being transcribed continuously. Okay? So if the transcription factor is FOXP3 in the nucleus, it will keep telling the regulatory T cell it will keep telling this T helper cell to make two major cytokines as the product. One is interleukin 10, the other is TGF beta. Okay, those will be the products coming out from here. If the transcription factor is T-BET, it will keep telling the T helper 1 to induce IL-2, TNF alpha, and interferon gamma. Okay, so these transcription factors regulate the products of these different kinds of T helper cells. Okay, does that make sense? Yeah.
Now, in terms of interaction, I don't want to get deep, but as a general concept, if one is activated, will it inhibit all others?
Not always.
Not all.
So these two can go side by side. But the amount of IL-6 will govern how much T helper 17 can be made versus regulatory T cells. We'll take a 3-minute break. I said I will stop. I didn't say I will stop completely. It was clear communication, your interpretation was different. Yeah. Based on its functionality. So immature tend to remain in the tissue. Mature, and the moment they get the antigen, they start moving towards the lymph nodes. While they're moving, they start upregulating the production of MHC class 1 and 2, so they do more antigen presentation. They increase the cell surface expression of co-stimulatory molecules. Yeah, so that's what I'm saying. Once they finish antigen presentation, they immature again. They become immature. So immature and mature, that's why we use the term antigen experience and non-experience for T cells, because this state doesn't go back. Once you have a helper cell formed, it doesn't go back to life. Okay, so it's there. However, there is plasticity in the system, because this is a T helper 17 because of ROR gamma T transcription factor expression. If ROR gamma T goes away and FOXP3 comes in, this can convert into this. So a lot of time your regulatory T cells can flip and flop. Okay. Yeah?
The cross-presentation between MSC class 1 and 2, is it, even though it's a bacteria, they may cross-present to MSC class 1? Or?
Yeah?
So it's like they do it just in case, huh?
Partly in case. It depends. Bacteria may be intercellular. Listeria is intercellular.
But like, for example, if it's extracellular bacteria,
Purely extracellular, no, not needed.
they don't do it. So they know it is I see.
It's pretty. It's a mechanism to kill T cells that could be cellular. Okay. So that's why we call it survival. So when signal 1 goes in, there is a reflex that comes and blocks signal 1. Okay. Signal 2 blocks that negative feedback, allowing signal 1 to continue. In the absence of this, the blocking signal that comes in kills the cell. So let's say it's a potentiating signal. Okay. So for simpler reason, keep co-stimulation. Okay. And signal 3 tells what kind of cell it should become. Now signal 3 is dependent on cytokine. If interleukin 12 is made, it will become T helper 1. If it's interleukin 4 made, it will become T helper 2. And why is it T helper 1 and T helper 2? Because they make a group of cytokines that have particular function. T helper 1 makes interleukin 2 to expand the cytotoxic T cells, TNF alpha to introduce fever, and interferon gamma to suppress viral production or protein production in general cells. And if a cell is infected by a virus, it cannot make viral proteins. You get what I'm saying? So this is the outcome of T helper 1. T helper 2 is more like allergic immune responses. So it will make interleukin 4, 5, and 10. 4 and 5 are required for plasma cell expansion. Okay. 10 is used for induction of IgE in particular. 5 and 10 both. Sorry. Yes, you have TNF alpha and TNF beta, but here we're talking mainly TNF alpha. Okay. Now, so this is just talking about the simple mechanism of how T helper 1 and T helper 2. But what makes T helper 1 make these three, and what it depends on, the cytokine induce a protein which is called a transcription factor. this transcription factor, this protein is now in the nucleus. As long as Tbet remains in the nucleus, it keep making T helper 1 cytokines to TNF factor. Now, based on that, different cytokines induce different transcription factors and therefore maintain the effector population as either T helper 1, T follicular helper, T helper 2, T regulatory, or T helper 1. You get it now? And therefore, as long as FOXP3 is on this T helper, it is called a regulatory T cell, and this regulatory T cell will keep making more of TGF beta and IL-10. If you remember IL-10, it is inhibitory for T helper 1. Therefore, this is quite useful to shut down immune responses, particularly T helper 1. Okay? Got it? Easy, right? Yeah. So let's move on to cell-mediated immunity, right? So your overall immune response is supposed to kind of protect you from bacteria, fungi, cancer, and even important in transplant organs. And all of these cells that are there are kind of relevant in that context, right? Where you have a high focus of T helper 1 supporting cytotoxic T cells and therefore involved in virus or any intracellular based protein or pathogen. T follicular helpers particularly target help in bacteria, T helper 2 for allergic responses, T regulatory generally suppress the immune response when it is not required in the body and also prevent autoimmunity. And T helper 17s are the ones that actually cause the autoimmunity. Okay? So you have all those cells present in your body. And when we restrict our talk to intracellular pathogens, for example, the HIV, you have two main effector outcomes. Infected cells can be killed by cytotoxic CD8 T cells that we described over there, which of course need help by CD4. And the mechanisms that they activate, they need more, in addition to just this mechanism, they also need to activate sweepers of the immune system. So once the target cell is killed, how is that apoptotic body going to be removed? There you have a role of macrophages that still need help through the CD4, and they also make more cytokines and interleukins to kind of support their functionality. In a global overview, you have the CD8 cytotoxic T cells, which are basically activated CD8 T cells that kill virus-infected cells and target all intracellular bacteria and viruses. The T helper 1s actually help macrophages, also help B cells to make antibodies against viruses, and also support microbes that they target basically again intracellular microbes, Mycobacteria, Leishmania, etc. T helper 2 switch to IgE mainly for allergic responses but also for helminth and parasitic infections. T helper 17, they are important in Klebsiella infections and fungal candidiasis because they actually enhance secondary neutrophil response. So the first neutrophil response comes by itself and secondary responses are kicked in by T helper 17. Having said that, they also are known to be there in arthritis. So all autoimmune arthritic diseases are primarily under T helper 17. T follicular helper go to making antibodies against all extracellular pathogens of all types, and T regulatory cells suppress it. Okay. Now, when your dendritic cells got activated and started moving towards the lymph nodes that we discussed earlier, how do they actually do that? You have the induction of CCR7, chemokine receptor 7, that starts taking these immature dendritic cells while they're maturing, start making them home towards the lymph nodes. While they're homing, there's increased antigen uptake in the phagosomes and they lose their phagocytic capacity. We discussed all of that. Within the lymph node, the CCR7, chemokine receptor 7, interacts with the chemokine ligand 19 and 21, which is present there. And this also induces more expression of MHC class 1 and 2 for them to primarily help in carrying out stronger antigen presentation. Now, when the antigen presentation kicks in, what happens in the context of a CD8 T cell? We discussed CD4 already, right? CD8 T cell again requires three signals. Signal 1 coming through the peptide MHC interaction and CD8. Then it needs signal 2. In signal 2, instead of having the CD28 and CD80 and 86 interaction, it primarily resides on the 4-IBB ligand on the CD8, 4-IBB molecule on CD8 that interacts with 4-IBB ligand on dendritic cell. This induction of this particular ligand is under the influence of T helpers that help the antigen presenting cell to upregulate the 4-IBB. And signal 3 is primarily the interleukin 2 receptor. Now, IL-2 is explicitly mandatory for T cell expansion. Okay. If IL-2 is not there, the clonal expansion shrinks. Okay. So this is why you need, while the T cell is getting engaged, CD8, on the antigen presenting cell through MHC class 1, it needs stimulates simultaneously CD4 T cell help. So this helps, so moment the CD4 T cell engages the MHC2 on the antigen presenting cell, while it is next to a CD8 T cell, it starts releasing interleukin 2 that is so important for the cytotoxic CD8 T cell expansion. Okay, is this clear? So this over here is now the effector function. Now, what happens to the cytotoxic CD8 T cell in terms of its effector functionality? The CD8 T cell that is now activated goes to the tissue, and in the tissue it starts looking for the virus infected cells. All MHC class 1 molecules are found on any nucleated cell type. So whichever cell is actually nucleated and infected is going to express on its surface MHC class 1. When it's infected, it will express antigens both of self and the pathogen antigen. Okay? So the cytotoxic CD8 T cell that is going and scanning around, it starts seeing a virus infected cell, uses the same T cell receptor that it used initially to get activated by an antigen presenting cell to look for the same antigen. When it finds the same antigen, it finds the target cell. Here, it does two things. It aligns depending on the direction. It engages, or it puts on its surface a receptor called, a ligand called FAS. Okay? And all cell types have on the surface called the FAS receptor. FAS receptor and ligand interaction induces apoptosis. So this cell, all your cell types on the surface express FAS. It is a death receptor. Normally it doesn't get engaged. So when the cytotoxic T cell comes, engages the peptide MHC, identifies the target cell, it engages also the ligand for the receptor on the target cell, inducing caspases and apoptosis. Apart from this process, it does another functionality. It releases in the vicinity two molecules. One are perforins and the other are granzymes. By the name of it, perforins perforate or make holes in the target cell. Granzymes that are released go through the perforins inside the cell and also start carrying out DNA damage, cutting the DNA short, inducing apoptosis. Now, cytotoxic CD8 T cell, one of them kills one virus infected cell, the next, so on and so forth, till it removes all the virus infected cells. And while it is doing it, it's very essential that these apoptotic cells that are dying, they need to be removed. So the same, and each time this engages, it releases in its environment interferon gamma and TNF alpha. Those basically activate macrophages to come and remove the debris that is coming out of it. Okay? So this is how the whole process kind of supports the removal of the target cells. Okay? Now in HIV, your cytotoxic CD8 T cell is actually not targeting an epithelial cell but a CD4 T cell. Okay? And that's what it starts doing. So over time, That's what happens in HIV. Now, this is just an overview that you can see for yourself later on when you are, frustrated after seeing Netflix, maybe. You can go back to this image and revise these. I'll stop here, and I just want to give you a little background on HIV now. So we use the concept of HIV to introduce to you what cell-mediated immunity is, what CD4 T cells are, what CD8 T cells are. Now, the HIV virus basically is a retrovirus. It gets incorporated into the cellular genome. And let me just, I like messing with your minds. Not too much, but not too less. Okay, here we are. You remember these transcription factors? Good. What do these transcription factors do? Give rise to cytokines. When are those cytokines produced? As a product of being activated, right? So each time this cell is getting a signal to make what? Interleukin 2 and release it, right? Just keep that statement in your mind. Before the virologists come in and kill you with virology, I just want you guys to understand the mechanistics. All proteins on the DNA level are under a promoter. Promoter means that this is where your transcription factors will come and induce gene transcription. Meaning they will start here to induce the synthesis of your mRNA. Agreed? Now the HIV is a retrovirus. It can go and get incorporated into the human DNA. Once it does that, once it's incorporated as a cDNA into your system, let's say it is downstream of interleukin 2. And interleukin 2 is going to be made in a large amount by your CD4 T cells. And that's the cell type that gets infected, right? So each time you get an infection, regardless of the sort of infection, you try to make interleukin 2 for this, but you end up making copious amounts of HIV. Okay? So when people suffer from HIV, you do not want them getting infected. So the disease has a very varying characteristic because we don't know where the virus actually gets incorporated. It may not get incorporated here, but somewhere upstream over here in a gene that is not going to be activated forever. And therefore nothing's going to happen. Therefore the progression of HIV disease is dependent on where it gets incorporated and how often that gene is being asked to signal. You get it? So some people have a very fast progression. For others, the latent phase can be as long as 10-15 years. But the moment people start having more infections, instead of giving protection, it starts inducing virus production. Okay? And therefore, you try to keep them, the people who have got immune compromise, you put them in bubbles. You try to keep them in an environment that is clean, no infections. And the idea is not to save them from HIV, they have it. The idea is not to activate the immune system to make more viruses. Okay? I stop here. Yeah?
What about the opportunistic pathogen, microbiota and these things? Are they susceptible as well?
So that's why you have to give them high doses of antibiotics and sometimes even chemoradiotherapy to shut things down. Okay?
So they can microorganisms free person?
Yeah, you need to be in a germ-free environment. Okay? So people which progress faster or slower is kind of mechanistically dependent on that. And my answer to your question, unless you understand, no, I'm talking to you there. So if you do not understand the mechanism behind it, although it's deep, but it's up to you. Okay? So I stop here. And if you guys, by the way, have some curiosity more after you go through the HIV session that's coming up in the next 10 minutes, if you want to, if it's still not clear, just let me know. I can have another session with you to explain things. If you don't want it, I'm happy. It saves my time.